Alright, so we're going to pick up now with our third section on our series on aerobic cellular respiration, and we're now at the point where we can start talking about the electron transport chain. And so having already seen something like this with our photosynthesis lecture, this should be a little bit less scary than it would be if this is the first time we saw something like this particular setup. So what we're going to do is we're going to walk through this, walk through it once relatively slowly. Normally in class I would go through this at least 2 or 3 times, but of course since this is recorded, you can go back and look at it as many times as you need to. So we'll walk through it fairly slowly, fairly deliberately. But again, this is one of those things, it's like telling a story, and you have to learn to tell a story. You have to know the process. It's not just memorizing a series of steps. You really need to be able to understand it to the point where you could even explain it to somebody else if you had to. Okay, so what we're looking at here, up at the top, we have, of course, mitochondria. Just to orient yourself, the top part of this picture, everything above here is the matrix of the mitochondria, so the core. Everything down here is the intermembrane space, so between the yellow and orange layers is the intermembrane space. And then, of course, this is the inner mitochondrial membrane. Okay, so built into the inner mitochondrial membrane is the electron transport chain. And so from our previous processes, we have our glycolysis, our oxidation of pyruvate, and our Krebs cycle, we have some molecules of NADH coming in. NADH is the reduced form of the electron carrier, so it's carrying with it electrons. The electrons are being dropped off here at our first acceptor, and we're also dropping off our H plus from NADH as well. If we go back and just really quickly review our look at our notes, we notice that with NADH, to produce this, we have NAD plus, H plus, and 2 electrons. So we have to keep track of those 2 electrons as well as the H plus. Right? So the NADH is dropping off the H plus, is dropping off the 2 electrons, and most importantly, it's dropping off the energy in these 2 electrons to the first acceptor. That's going to release NAD plus back into our earlier steps. Again, Krebs cycle, oxidation of pyruvate, and glycolysis. And from that point on, these 2 electrons are going to pass through the first series of acceptors in the electron transport chain. So these 2 electrons go from here, eventually down to here, and you'll notice the arrow pointing down. What this represents is these electrons are giving up energy. They're actually losing energy as they go from this first acceptor to the second acceptor through this shuttle. Now the energy they give up is being used to move hydrogen ions from the matrix into the intramembrane space, from the inner compartment to the outer compartment. And this is active transport, which is why it requires energy. The energy from the electrons is used to move H pluses from the matrix to the intramembrane space. We're moving H pluses from where they're low concentration to where they're high concentration. Alright, so from that point the electrons go on to our second electron acceptor. They're going to give up some more of their energy. They're going to move H pluses again from the matrix to the intermembrane space. So again, they're doing active transport of H pluses from the matrix to the intermembrane space, active transport using the energy from these 2 electrons. And then eventually these electrons end up with a third electron acceptor, initially at least, where they give up the last little bit of their energy, or almost the last little bit of their energy, moving H pluses again from the matrix to the intermembrane space. And at that point, the 2 electrons get paired up with a half of an O2 molecule, 2 H pluses, and that's going to join together to form water vapor. All right, and so when you breathe out, one of our products of cellular respiration that we talked about back again on our notes page was water vapor. Right, so when we breathe out, there's the H2O that we're breathing out. It's coming from, oops, that's the wrong lecture. It's coming from this water vapor that's produced in the mitochondria doing aerobic cellular respiration. Okay, so we have one of the functions of this process is to actually help get rid of H pluses. And so if we go, well actually let's go back to our notes here for a second, and I just want to show something here really quickly. If we talk about what aerobic cellular respiration accomplishes, it helps us to get rid of H pluses, right? It prevents the mitochondria from becoming too acidic. By getting rid of H pluses, not only does it create ATP, not only does it recycle the NAD plus back to glycolysis, etc., but doing this process helps us get rid of extra H pluses, which otherwise would make the mitochondria very acidic, causing all kinds of things to break down. Those H pluses pair up with electrons and oxygen to form water. Alright, so what have we done so far? We've taken our NADH, we've oxidized it, we've moved electrons through an electron transport chain to power the movement of H pluses from the matrix to the intermembrane space through active transport. Alright, we also have FADH2 coming in. What does FADH2 consist of? Well, it consists of FAD, it consists of 2 electrons, and it consists of 2 hydrogen ions. So FADH2 is going to come in a little bit later than NADH, and there's actually a very important result or implication of this. This FADH coming a little bit later is that it's going to skip the first electron acceptor. So when this FADH2 gives up its 2 electrons and FAD and 2 H pluses, it's at the second electron acceptor. And again, same as what we saw for NADH, Giving up those electrons and their energy allows for the active transport of H pluses from the matrix to the intermembrane space, from the second electron acceptor and the third electron acceptor only, but not the first one. And then again, the electrons from FADH2 end up as part of water. FAD gets recycled back to the Krebs cycle, and H pluses are moved against their concentration gradient into the intermembrane space. Now, I mentioned a second ago that FADH2 skipping the first acceptor has some important implications. One of them is that for each of these NADHs that come in, that's going to create a total of 3 molecules of ATP. Look how many electron acceptors we're hitting right here. 1, 2, 3 acceptors. NADH gives us 3 ATPs per molecule eventually. FADH2, on the other hand, because it only goes through 2 electron acceptors, will only give us 2 molecules of ATP. So that's really important as far as how much each of these two energy currencies is to the cell, NADH versus FADH2. Each one of these NADHs is worth 3 ATPs. Each FADH2 is only worth 2 ATPs. Okay, so that's important as well. Now, if we go all the way down here to the end, there's a very special enzyme called ATP synthase. And ATP synthase is a transport protein. What this is facilitated transport of H pluses down their concentration gradient. Now, what we've done throughout this entire series of electron acceptors, the electron transport chain, moving H pluses from low to high concentration, using energy from electrons coming in from NADH and FADH2, what we're doing is we're setting up a very, very steep concentration gradient. And these H pluses really want to get out of the intermembrane space. They really want to go back into the matrix. All right, so one of the reasons for that is concentration. There's a high number in the intermembrane space, a low number in the matrix. They want to go from high to low concentration. So that's point number 1. Point number 2 is there's also an electrical gradient. All these H pluses inside of here repel each other. And so they want to get away from each other for that reason as well, not only because there's a high concentration, but because they're all repelling each other. So they really want to get back into the matrix, but the only way they can do that is if they pass through ATP synthase first. And so as these H pluses move through ATP synthase literally spins around in a circle. It's a kinetic energy. It spins around in a circle, and that spinning creates the potential to join ADP and phosphate together to form ATP. So as these H pluses pass through ATP synthase, this molecule spins around in a circle, and that creates for the potential of ATP production. And that's where the bulk of our ATP comes from inside the cell. It's the result of the electron transport chain, all these blue guys coming in, as well as ATP synthase doing what we call chemiosmosis. So when these H pluses move through ATP synthase to create ATP, we call that process chemiosmosis. Right? So that's a long time on one slide, but there's a lot going on in this slide. This is basically, a good chunk of the chapter is shown on this slide. All right, so what we're doing there again is we've moved the H pluses from the matrix to the intermembrane space using the transport proteins to do active transport powered by the energy and the electrons that have come in from NADH and FADH2. Now, one thing I want you to think about is we talked a lot about how these electrons have a lot of energy. These electrons are giving up a lot of energy. Where did the energy come from? Well, where do the electrons come from? The electrons are coming initially from glucose, right? And glucose is a high energy molecule. We know that. But why is glucose a high energy molecule? Well, glucose was created using sunlight. And so the electrons in glucose are high energy because the energy was put there by the sun. As we extract the energy from those electrons, that allows us to make some ATP. Right? As the H pluses move down their chemical gradient from high to low concentration, H pluses also repel each other, so they're going to move electrically as well. And that creates this movement through ATP synthase, which generates ATP. And again, that's the joining of ADP and phosphate, adenosine diphosphate, and phosphate together to form adenosine triphosphate. This is a different picture, different book, different illustrator, but the exact same thing going on. All right, so now we have, outer membrane, intermembrane space, inner mitochondrial membrane, and matrix. We'll go through this a little bit faster. All right, so the NADHs come in, they're carrying these 2 electrons. These 2 electrons have energy. And so when the electrons are dropped off here, we also recycle NAD plus back to glycolysis, oxidation pyruvate, and Krebs cycle, and we also release H pluses. These electrons, as they give up energy, allow for the movement of H pluses from the matrix to the intermembrane space through a series of electron acceptors. Eventually, those electrons from NADH end up with H pluses and oxygen to form water. Meanwhile, we also have FADH2s coming in, dropping off electrons. Those electrons power the movement of H pluses again from the matrix to the intermembrane space, filling up the intermembrane space with H pluses. Those H pluses really want to get out because of a concentration gradient and electrical gradient. So as the H pluses move through ATP synthase, which is their only route to get through this membrane most of the time, the H pluses make this ATP synthase spin in a circle, and that spinning around allows for ADP and phosphate to join together to make ATP. And again, that's where the bulk, roughly 32 of the ATPs of the total come from, is from this chemiosmosis. All right, so now we see this picture again. It starts to make a little bit more sense, and you should be able to follow through this. If you can't follow through it, this is a good point to ask questions, because chances are other folks in the class don't understand it either. So work through this, be able to explain it, be able to look at this picture or the one that came before it, this guy here, and follow through it. And once upon a time, there was an NADH, and eventually ATP lived happily ever after. There's a whole bunch of steps in between that you need to be able to kind of track to make sure you understand this all the way. Okay, so that's the bulk of the step-by-step aerobic cellular respiration process that we're going to look at. We're going to take a look now at some theoretical stuff, some energy stuff. All right, so here is the overall equation for the aerobic breakdown of glucose to CO2 and water, and of course ATP. That breakdown of glucose should yield about 686 kilocalories per mole of glucose. That's the energy output from one glucose, is 686 kilocalories per mole of glucose, right? Now, this is a negative number, and because it's a negative number, what this means is as you go from reactants over here to products over here, we are giving off 686 kilocalories per mole of glucose. So that makes this an exergonic process because energy is being given off. In a cell, that number can be even higher. They can actually give off more energy. They're a bit more efficient than what we can do even in a lab setting. So the number can actually be a little bit higher. One thing we also want to think about here is other forms that energy takes as this reaction proceeds, or as these reactions proceed. Obviously, we're getting some ATP out of this. That's the whole point of doing it. But we're also going to get a fairly substantial amount of heat. And so the reason why this process, from beginning to end, from glycolysis to chemiosmosis, is so complex is that you have to release the energy in small steps. Because if you didn't, if you release the energy all at once, that's actually enough energy to cause the cells to overheat and maybe even burst into flames because the energy amount is so high. So we want to release the energy in small steps. It's kind of like the gas in your car's gas tank, right? It's a huge amount of energy. If you release it all at once, it's going to be fairly catastrophic for you and your car. So you want to take the energy in the gas, release it in very, very small steps, very, very fine spray and fuel injectors in your car's cylinders, not all at once, but in tiny, tiny, tiny steps to extract the energy safely and efficiently. Right? So the reason why this process is complex is it's safety, basically. The energy has to be released in small steps so the cells don't actually overheat themselves. So here's what we've done so far. Glycolysis, we get 2 ATPs, we get 2 molecules of NADH. Each NADH is worth eventually 3 ATPs each, so 2 times 3 is 6 ATPs, and that's again through chemiosmosis. We oxidize pyruvate. Again, we don't get any ATPs here. If we go back to our notes, we notice that oxidation of pyruvate, if you remember, did not generate any ATPs at all. Oxidation of pyruvate, which is right here. We get CO2, NADH, acetyl-CoA, but 0 ATPs. Oxidation of pyruvate gives us 2 NADHs. And again, each one of those NADHs is worth 2 ATPs each. 2 times 3 again is 6 ATPs total from chemiosmosis. We go to the Krebs cycle. The Krebs cycle gives us a total of 2 ATPs directly. It gives us 6 NADHs, so 6 times 3 is 18 ATPs there. There's a big payoff right there, is those 18 ATPs. And then FADH2 is worth 2 ATPs each, so 2 times 2 is 4 ATPs. So if we add up those numbers, theoretically, there should be 38 ATPs produced. That is if you were a eukaryotic cell. I'm sorry, a prokaryotic cell. Prokaryotes produce 38 ATPs total. Now, the number itself, the actual number is lower. If you remember back in the beginning, we talked about glycolysis and how sometimes you have to spend energy to make energy. So we had to spend 2 ATPs. We got 4 ATPs back. So really what this means is that theoretically the total ATP yield is 38, but it costs us 2 ATPs to get going, so the actual yield is 36 for a prokaryotic cell. 34 for a eukaryotic cell, because eukaryotes have to do that step of actively transporting pyruvate into the mitochondria. So for us, we have to spend 2 ATPs initially for glycolysis. We have to spend 2 ATPs to get pyruvate into the mitochondria. So 38 minus 4 is 34 ATPs. So we say then, for a eukaryotic cell, we get 34 ATPs per glucose. For a prokaryotic cell, theoretically, we get 36 ATPs per glucose. The actual numbers might be a bit lower for reasons we'll talk about later, but theoretically, at least, that's how the numbers should shake out. Okay, what happens if you don't have glucose available? What happens if you're in a situation where glucose levels have gone down to the point where we don't actually have enough to do glycolysis, etc.? Well, we have some fallback options. There are other kinds of molecules that we talked about all the way back in chapter 2, which can also serve as energy. So we can use fats, we can use nucleic acids even sometimes, we can use proteins as an energy source if we don't have carbohydrates available. So to do that, the mitochondria only deal in the currency of the things we talked about so far: glucose, pyruvates, acetyl-CoA, those kinds of things. Mitochondria only know how to use those things. So if you're dealing with fat or protein, for example, you've got to take those molecules and convert them into something else first before our mitochondria know how to work with them. Okay, so that's a key step, is converting fatty acids into acetyl groups, or converting proteins into various things like glucose that can be used by the mitochondria. So we'll see those as well. So what happens if you don't have glucose available for the relatively short term? The first thing you're going to do is your body's going to fall back on the catabolism of protein. Catabolic just means you're breaking down something. Anabolic is building, catabolic is breaking down. So here we're breaking down proteins, right? So if we break down a protein, we know the building blocks of proteins are amino acids. And so what we have to do with amino acids to use them in this context is to take the amino acids and deaminate them. We're going to remove the amino group, and what's left behind after we remove the amino group is something which can be used in the mitochondria to produce some ATP, or in glycolysis, possibly as well. All right, so the amination, or the deamination step, I should say, one of the downsides to that is that when you deaminate the amino acid, that amino group actually becomes ammonia. And ammonia is relatively toxic to cells and toxic to your body unless you detoxify it first, and that actually takes place in your liver. So you do have a way to break down the ammonia that's produced. It goes to your liver. The ammonia gets converted to urea, and then eventually your kidneys filter urea out of your blood, and it's excreted in the urine. So it does have an outlet for the body when you break down proteins this way. What's left of the amino acid after the deamination step is then used in glycolysis, or the Krebs cycle. So for example, alanine is an amino acid. Alanine is converted to pyruvate that of course gets transported actively into the mitochondria, where it gets oxidized and enters the Krebs cycle, etc. Aspartate is another amino acid which can be converted into oxaloacetate, which is a part of the Krebs cycle. So it's going to feed in there. Now notice these molecules aren't going through the entire series of steps we talked about that glucose does. Converting proteins to ATP is not as efficient as using carbs, but it works. It keeps you alive long enough so that you might be able to find carbs again at some point in time and use those to run cellular respiration to produce ATP. Fats are a little bit different. What happens with fats is they're broken down to fatty acids and glycerol, which are kind of the main components of a triglyceride molecule, for example. The fatty acids are then converted to acetyl groups through a process called beta oxidation, which is also an aerobic process. And we know what happens to acetyl groups. They are the end product of the oxidation of pyruvate. They are going to feed into the Krebs cycle, and these acetyl groups are no different. So we're going to feed these into the Krebs cycle, and that's going to give us an additional source of ATP, which you can use even longer term than proteins. The thing with fats is they're also very efficient. You can pack a lot more calories into a volume of fat than you can of protein or carbohydrates, so they last a while. They're roughly 20% more energy held in a molecule of fat than there is in a molecule of glucose. So they're very, very good at storing energy for the very long term and not taking up a lot of space either. All right, so here's kind of an overall look. These pictures seem like they're getting more and more complex, but this is really everything we've seen so far. If you look right down the center of this column here, starch and glycogen get broken down to glucose. Glucose goes to glycolysis to produce pyruvate. Pyruvate goes into the mitochondria to get oxidized into CO2, and the acetyl groups, which become acetyl-CoA, those go to the Krebs cycle. That gives us our CO2, ATP, NADH, FADH2. Krebs cycle eventually runs into the electron transport chain, where we use chemiosmosis to make ATP by using oxygen and also making water in the process. Right? So this is what we've been doing for most of the chapter. With the proteins, we take the amino acids, we deaminate them, those become waste products down here. And then depending on which amino acid you're looking at, those products can then feed into glycolysis, oxidation to pyruvate, Krebs cycle, or somewhere along the line where it can be eventually turned into ATP as well. Fats are broken down to glycerol. Glycerol eventually becomes pyruvate, etc. Fatty acids feed into the Krebs cycle as acetyl groups, and so that's going to give us ATP as well. So this is a really big overall picture of what we've seen so far, using not only carbohydrates, but also proteins and fats to make some ATP. All right, so our last section we're going to deal with deals with fermentation. So I'm going to stop here, and I will start up again with a fourth section on anaerobic respiration.